Hello, my name's Paul Carney. I'm a professor here at the University of Florida. I'm also the Wilder Chair for Epilepsy Research at the University of Florida. I'm a clinician as well as a researcher in epilepsy, and I came to the University of Florida in 1998. I had done my training in Cleveland, Ohio, at Case Western Reserve University and Rainbow Babies and Children's Hospital. Then I subsequently trained in neurology, epilepsy, and sleep medicine at the University of Michigan in Ann Arbor, Michigan. When I came to the University of Florida, we actually started the program. It was an epilepsy program, and it became very clear to me that we needed to do more than to just treat patients. And so during the last 10 years, I've had the good fortune of being able to assemble a fantastic team, a team of postdoctoral fellows and young scientists. These are PhD scientists and master's students and undergraduate students who've come to the University of Florida specifically not only to learn, but also to make a difference. The major goal of the research being done in our laboratory is to understand the mechanism underlying epilepsy and to apply this knowledge toward developing new treatments. Treatments that are going to be more meaningful and effective. We've taken what I refer to as a unified cross-disciplinary approach to tackle the fundamental epilepsy problems from a clinical, biological, and engineering standpoint. So the first area is called optogenetics. Optogenetics combines gene therapy with local optical stimulation. In this method, light-sensitive proteins are introduced into the brain where focal seizures originate. The activity of these neurons can then be inhibited by simply shining yellow light onto the neurons, or they can be activated by simply shining blue light onto these neurons. The approach allows for very specific targeting of neurons and to minimize side effects. Now, by implanting electrodes into the brain, seizures can be detected and then quickly suppressed by a very small light source implanted in the same area. This approach can be combined with existing seizure prediction and detection techniques in order to prevent the seizure just before it would occur. With further development, we believe that optogenetics will be able to prevent seizures without the patient having to resort to drugs or to the resection of a portion of the brain. A second area focuses on understanding the evolution of epileptogenesis or development of epilepsy using state-of-the-art technology. It is important to study how and where seizure activity originates and subsequently spread in order to better define the seizure focus. Work currently is being done to characterize brain connectivity changes both in an animal model of epilepsy and in patients with epilepsy, and structural brain connectivity changes we believe will subsequently be correlated with functional measures such as cognition or learning and memory. A third area of research focuses on developing novel drug delivery tools. We are conducting studies to understand fluid flow in the extracellular space in the brain in order to optimize drug delivery directly into the brain. Direct applications of therapeutic agents would enable unparalleled control of drug dosing, spread, and targeting. The final area is based on optical imaging. We believe that this novel optical imaging tools will significantly advance not only understanding of brain networks, but will also lead to more accurate mapping of the epileptogenic focus, both for epilepsy surgery and also for knowing where to place very specific devices that will eventually control seizures. Finally, I'd like to sort of end by telling what I consider probably one of the most remarkable stories that I've heard in recent years. We had the good fortune, actually the blessing, of being able to take care of a patient, Rob Batzel, who I got to know when he was a young man, a teenager. And Rob, of course, will tell his story, but Rob became very touched and I think very interested in epilepsy, not only because he had seizures, he's very well controlled now, but because he saw how others really weren't as fortunate as he. And he became very interested in research. We invited him, he visited our laboratory together with his spouse Brittany, and decided to form the SOS Foundation. And so we are very fortunate to have someone with such a deep understanding of the disorder who is willing to support epilepsy research here at the University of Florida. On behalf of my lab, my students, my postdocs, and my colleagues, I want to thank Rob and Brittany for their efforts in partnering with us to really work on this very important disease, and hopefully we'll have a cure for the disorder in the years to come. So thank you again, Rob, and thank you again, Brittany.